Good morning. My name is Bryan Lewis. I'm the president of KidneyCAN. I want to say a deep, heartfelt thank you for traveling from across the country and different places to be here today. It's exciting not only for the kidney cancer ecosystem, but for patients out there who are listening in. We really appreciate the smartest people basically around the globe coming together to figure out how to accelerate cures. So thank you. Before I get to a few housekeeping points, I just want to say thank you to the KidneyCAN team that made this all possible. My good colleague Susan Poteet back in the back here is instrumental in setting a lot of this up today. Couldn't do it without her. Also want to shout out to Tony Schwery and Hans Hammers, who are our executive committee members and helping to shape some of the content. Thank you, Tony and Hans. Finally, want to say thank you to the KidneyCAN team, who's not in this room. They're out there doing all the things. A couple of years ago when we had our inaugural KCRS, there was Susan and me and a handful of volunteers. Now we have a team, and so really appreciate all the work they're doing. So thank you. And we couldn't be here also without all of our sponsors, and you'll see some of the banners around that recognize them. So thank you to all of our sponsors who've made this possible. As many of you know, the backbone of the KCRS is the Department of Defense research funding for kidney cancer research. And many of you also know that in 2016, we had $0 dedicated to kidney cancer research. Thankfully, today we just got, we found out the House this summer passed $50 million for kidney cancer this year. So we're gonna keep driving that train in Washington to continue to increase funding so that we can bring more bright minds into this ecosystem and figure out how to accelerate cures. So thank you for that. And I want to thank all the patient advocates and many people in this room who have advocated in Washington virtually and in person to make this possible. So thank you for that. Last sort of general comment is we really want to encourage collaboration. We want to encourage dialogue. We want you to be thinking outside of the box. We want you to be sharing and breaking down silos. So please, within the confines of mask wearing when you need to, break down those silos today and tomorrow. Housekeeping, your microphone's on your table. There's a little face of a person speaking icon in the middle. That has to be pushed in order to speak. We'll try to help you out. We have a couple of folks that'll be running around with handhelds. If you have a question, you can raise your hand. I think many people know the restrooms are sort of behind the wall back over there. Self-serve food all day and drinks. Feel free to get up and go and come and get something to eat. Slido is the question and answer capability online that you have a little tabletop in front of you. You can scan the QR code. We're gonna have a lot of fun tonight, I hope, at Estia, where we went a couple of years ago. Have some food and drinks. Starts at 5:30 if you want to have a cocktail, 6 o'clock if you want to eat, a little of both. And then I got a plug for, where's Bradley McGregor? There he is. Brad was kind enough to volunteer 2 years ago. He and Monty Powell ran from the Royal Sonesta Hotel up to the Art Institute and up the Rocky Steps. We sort of taglined it, the Rocky Run for Ralph, who Ralph Knapp is our co-founder for KidneyCAN. He's going to be here at some point, I think. And, Bradley has agreed to sort of head a team that's going to run tomorrow morning at 6:30 from the Sonesta Hotel. Any runners out there, time to go out and buy some sneakers tonight. Meet you at the Sonesta tomorrow morning at 6:30. And on that note, I'm going to turn it over to my esteemed colleagues, Tony Schwery and Hans Hammers, to introduce our fireside chat. Thank you, everybody.
Start with a new meeting better than two of my colleagues and friends. Well, it's the first meeting back, so. Can you hear me now? Yeah, can you hear me now? Very well. So thank you again. Can't start a new meeting live, with better folks here. My good friend Hans Hammers and two of my mentors, colleague and friend, immunotherapy power couple, Arlene Sharpe and Gordon Freeman. So thank you. And I understand from you that this is the first meeting almost live. So we're all in this together. So people are very familiar with your work, okay? And we solicited many questions. We wanted to make this as a fireside really chat and as relaxed as possible. And you should see the type of questions that came that, I'm gonna just take some of them, but I'm gonna start is why immunotherapy? And, immunotherapy in cancer at least had some rough times. I remember my first mentor telling me, if I wanna go through something, probably not for the time being immunotherapy. So how did you start, each one of you, by immunotherapy and your experience before the explosion with the immune checkpoint inhibitor? Maybe starting with Arlene.
Okay. So I guess my background is one where I was studying virus-host interactions as a graduate student. I worked on reovirus pathogenesis and got very interested from the perspective of the immune virus dialogue. I also became interested in pathogenesis in general, and that led me to do a residency in pathology. At that point in time, I would see cancer, I would see these infiltrates, made me very interested, but I knew that there needed to be more basic science. And so I guess one of the things that is a theme throughout my career is really genetics. I did viral genetics as a graduate student. I went to Rudolf Janish's lab as a postdoc, made knockout animals, and now we use CRISPR approaches. And so that really enabled me to sort of study molecules at a time that they were just emerging with molecular biology. And I was very fortunate to have one of the best molecular biologists to collaborate with in terms of identifying molecules. And then I would say, gee, how important are they? And we'd knock them out. And I think that is sort of where my really travels in immunology went. And, looking at different molecules, identifying together, really working together on families of molecules. And that really understanding inhibitory molecules led to questions related to tolerance and cancer.
And Gordon, I know you were with famous immunologist Lee Nadler, stemming also with your paper. You're very important, I would say the most important paper in the PD-1, PD-L1 pathway story in the Journal of Experimental Medicine. But I heard you went also through virology and translated that to cell line, cancer cell line.
Yeah, as a graduate student in virology, I had learned molecular biology. And at that point, immunology was factors and reactions and no molecular identifications. So I brought molecular cloning to an immunology lab. And in that lab, they discovered an antibody called B7 because it was the 7th molecule on B cells. And we cloned that molecule and found it activated and stimulated T cells. It provided this critical second signal for T cell activation. And we took that molecule and put it into cancer cells, and it activated the immune response against cancer cells. So we thought that was going to be a successful immunotherapy, but it turns out not to translate well. But we went and looked for what molecules looked like B7. And this was the dawn of the sequencing of the genome. And there were suddenly 20,000 genes to look at. And we found a molecule that looked like B7. And we now know it as PD-L1 because it's the ligand for PD-1. And we discovered that turned off the immune response. And then our first hint of where it came from was it came from an ovarian tumor. So we went, which is a surprise because you don't expect to see an immunological molecule coming from a tumor. And so we got our pathologist, David Dorfman at the Brigham and Women's, to use our antibody and look by immunohistochemistry for where was the molecule expressed. And it was expressed on the surface of tumor cells. And we were collaborating with Dr. Hanjo at that point and told him that it was on tumor cells. And we continued our work in the humans, showing that the PD-1 was expressed on activated T cells and that blocking the interaction would enhance immune responses. And he did mouse models which showed if you block the interaction that you would get an anti-tumor response. And that's how PD-1 therapy has evolved.
I mean, a beautiful story. And the rest, I would say, is history with how PD-1 and PD-L1 inhibitors, just transformed in the fourth wave of anti-cancer therapies. My friend here from Dallas telling me that, science is great, but you want to ask more personal questions. So ask yours.
Sounds good. So it's always exciting to actually have a married couple to go into science together, and it seems like you've been collaborating so much. How did you actually meet? How did that happen? And he would ask, was it love at first sight?
This is broadcasted international.
It was attraction at first sight.
Attraction.
We actually met in German class as undergraduates. Both of us thought that if you were going into science, German was the language to study. So we met in German class, not in a science class.
And it was an early morning class. And I, at that time in my life, was not an early morning person because I'd worked late in Don Wiley's lab trying to do structures. And I found that I could go to Arlene's
And so Gordon would show up in my dorm and say, so what was the assignment? Do you have notes? What's on the test tomorrow? And then I realized he kept coming and like there wasn't that much to ask, but he was interested in more than just was going on in the German class.
Yeah. I would guess you don't speak any words of German.
I've forgotten most.
No, I mean, this is great. One question that came, and then we'll give it to the audience with anything you want, the mic, is during this pandemic, everyone got a new hobby or something new. So I don't know, Arlene, is Gordon the best cook or what? What's your hobby? What's your hobby?
Well, I think when we were graduate students and postdocs, we divided cooking and labor pretty evenly, but during the pandemic, I think it's changed.
I always tried to work in my office, and home was a place to relax. But with the pandemic, things blurred between work and relaxation. And actually, I rediscovered cooking. That was my outlet and relaxation and creative outlet during the pandemic.
I mean, that's great. Want to take any questions, please, from the audience, or I will continue with personal questions to our guests here. Perhaps anyone? Please, Lisa.
So if you could fast forward, if you could fast forward 5 years, where do you think we're going to be? What will be the kidney cancer and immunotherapy?
I think combination therapies are what's going to be developed. Things like Nivo and VEGF inhibitors now are first line. I think the clinical trials with additional combinations, it's just amazing. There are over 300 different agents that have been shown in a mouse model to improve immunotherapy with PD-L1 or PD-1 blockade. These are all advancing through the clinic. I think the critical thing now is to prioritize the clinical trials, identify what's working, and I think some of these are going to succeed and really improve kidney cancer therapy.
Yeah, I would agree. I think combination therapies, but I also think how we get there is going to be important. That I think we have all of these omics and other approaches that are identifying molecules and pathways. But now I think bringing it all together with an integrated understanding, spatial approaches, we're beginning to have an understanding that different tissues are different. What is the difference in the microenvironment in the kidney versus a metastatic environment? And understanding that, I think, will help us be able to think about prognostic, diagnostic, and new types of combinations.
Perfect. Any other questions here? Ari, please.
So that was an awesome story so far. I'm curious your thoughts on the role of the host in all this. So, when I say that, I mean the implications of immunotherapy on different HLA types, whether that's with response or with toxicity, something that and your thoughts on whether, by country or different responses, all these things, how they may interact, and whether that should be something that is regularly looked at.
Well, I think that's an interesting question. We're thinking about benefits and then immune-related adverse events, for example, which is really an area that's just unfolding. Why it is some people experience them, what's different. I think genetics is part of it, how similar these are to autoimmune diseases. But I think if you step back also and think about the host, how does age, how does obesity, and the microbiome, we're beginning to get that there are differences there, and we really need to understand what differences are in different host microenvironments and how that impacts immunotherapy, and are there new combinations that suggests?
We have an audience question, which is there a PD-1 minimum threshold for effective response?
Do you mean the amount of drug delivered or
Maybe we can look at-
expression of
Yeah,
PD-1?
maybe both. We can maybe.
Yeah,
touch-
the expression, as
Expression
a
policy.
clinical trialist, in PD-L1
Yeah.
negative tumor like kidney cancer, we've seen responses. So this question is probably a natural question, knowing the way to detect how much PD-1 and PD-L1 there is in human tissue is not that sensitive. So from the lab, do you think you need a minimal expression somewhere for the ligand or at least a receptor for an immune response?
Yeah. I think the PD-L1 blockade works both if you have PD-L1 on the tumor, and it's also critical in like the lymph node in interactions between the antigen presenting cell and the T cell. The assays of PD-L1 are being improved and compared and quantitated by computer now, and all those assays are being incrementally improved. It's still immunobiology, which is a little inexact and fuzzy.
I think that's perfect. One of the questions here, please.
Can I ask a question? I'm sorry. I'm over here. The microphone.
Okay.
Oh, sorry.
Oh, yeah.
Yeah.
Okay, you're first and please.
I'm sorry. Very exciting, of course. There's so much therapeutic combination therapy developed, but I want to get your idea strategy, how to find out who is going to respond and who's not. I think all of this discussion is about that issue. Because when you combine all therapy together, it seems like and give it to everybody, it seems very inefficient to know who is responding. How do you select people, stratify that would be best responder for this therapy or that therapy? To find out, the bottom line is who can get the most benefit out of a treatment. So I want to get your idea of how to go after and find out who might be the responder for immunotherapy and maybe for other TKI therapy, specific therapy, because I think they might be specific responder to specific therapies, and not all comers are going to respond the same.
I think you raise a really good point. I think we're going toward more personalized immunotherapy, if you will. And I think transcriptomics provides us with a great opportunity to study cohorts of patients who are initial responders versus those who never respond, or those who respond initially but then progress. And being able to understand those and what are differences there can suggest potential biomarkers and new targets, and help us understand individually what are biomarkers and targets for identifying response and what to look for, and then to predict whether a particular patient has a certain profile to be a responder, and then what to choose.
I think this is great, and many of us are working now that clinical trials capture tissue in good shape, and I think we will be able to get there. The issue is responder, non-responders, sometimes the truth in the middle, and you don't have curves that look like this. But I think biomarkers is very active area of research. It's one of the priorities on every agenda. One suggestion, since we have a large online also participant, is the person, please state your name, and I'll be happy to repeat the question. But I would love one of our patients. We're lucky here that this is a horizontal approach. We have a lot of patient and patient advocates. So one of you, Susan, I don't want to put you on the spot, but from a patient and a patient advocate, what would you ask Dr. Sharpe and Dr. Freeman? What are you interested in most?
Cures. It's remarkable. I can't tell everybody in the room how much it meant to us to meet the first IL-2 complete responder. And no, some people didn't die in 2 years. And we have more of those every year. As you said, we're going to need more therapies, more targets, more options. Will we ever get to 50% complete responses?
Hopefully one day, and one day soon. Any other question, perhaps from, oh yeah, Jovan, please, go ahead.
So my question was, what's your opinion on cell therapy and combination of immunotherapy and cell therapy, renal cancer, and other solid tumors?
So cell therapy for the audience and immunotherapy. Thank you. In solid tumors and kidney cancer.
I think that there's opportunities there. I think cellular therapies and solid tumors are really just beginning, but I think it provides a different avenue because you can manipulate T cells, for example, in a different way, in a different state, where you can selectively target those cells in a certain way and then combine it with PD-1, for example, thinking about the type of cell, how you can rewire the cell to get a more durable response or overcome different types of resistance. So I think there's going to be a combination opportunity there that should be really exciting.
I think the success of CAR T in leukemia has really excited people. And there's now a tremendous amount of creative effort being made to bring CAR T to solid tumors. There's a number of problems to be overcome, but there's a lot of creative ways people are testing how to bring CAR T to solid tumors. There's some good possibilities for targets in kidney cancer that are being explored, and really trying to tune the CAR T so there's good anti-cancer efficacy with little less toxicity.
Perfect. Dr. Zakaria Youssef from University of Iowa.
Yeah, thanks, Tony. It's a great conversation. What other immune receptors excite you beyond PD-1 and PD-L1? And
This is a great question.
That's a setup for me.
That's a setup. So for the audience, other checkpoint or other receptors that excite you.
Again, I work on something called the B7 family, which is the critical molecules that up or down regulate an immune response and include PD-L1. So we went and looked for other cousins of these molecules and noticed that in kidney cancer, there was a lot more of one called HHLA2. It really went up a lot in kidney cancer. And we looked again and again, it can be expressed on the kidney cancer cells. Now, the problem was people didn't know how it worked. So we went and recently identified that the interaction of HHLA2, it has an inhibitory receptor that turns off the immune response. Sounds a lot like PD-L1, PD-1. And so we're hoping to develop this as an alternative immunotherapy. And the other exciting thing was that when we looked at kidney cancer, there were kidney cancers samples which Sabina Signoretti showed had PD-L1, and that was a modest percentage. And then there was very different patients whose tumors had HHLA2. So we're really entertaining the idea that this is an alternative immunoevasion pathway in kidney cancer. And now we want to see if we can safely and effectively target it. The other targets that are people have enthusiastic about is, I think TIGIT has now advanced pretty far in the clinic and is in phase 3 trials. And we'll see the earlier trials have worked pretty well. And we'll see whether TIGIT as a monotherapy or combinations with PD-L1 or PD-1 is successful.
Perfect. Perfect. Dr. Zhang from UT Southwestern, please use the mic.
Thank you. So I'm just wondering, everyone's talking about the TKI plus IO therapy. I'm just wondering whether the treatment, sequence of treatment will make a difference in terms of efficacy of patients, especially in kidney cancer. Because anti-CTLA-4 is prime consequence, right? PD-L1, of course, is not as hard like kidney. But in terms of VEGF and inhibitor coming in place, the order of treatment, does that matter? Or does that make a difference?
So this is a great question. You know, we know that TKI, VEGF, TKI, and PD-1 inhibitor are standard now in first line, one of the standards. So do you think sequencing make more sense, at least preclinically together? And, you know, some of the work you've done through CHIME about KDR, VEGFR2. Arlene?
Yeah, so I think we've done work to understand durability of responses. And what we found with PD-1 is that if you block it early, most of the work has been in infection models, but a very talented fellow in my lab, Kelly Burke, has translated this to tumor models, that if you block PD-1 during priming, you can get an early, very exuberant response, but you get short-term rather than long-lived memory. And so that would suggest with combination therapy, you would want to use something else to prime and then add PD-1 for the combination. And indeed, those are the types of things that we are testing now together. And so I think combinations and timing are very important. Of course, I think a tumor microenvironment is one of the most complex because you can have memory cells there, you can have exhausted cells there, you can have prime cells there. But if you have an environment where you're going to want to have a desert or something where you're going to want to bring in new cells to activate them for the first time, then you may want to activate them with something and then add anti-PD-1 shortly thereafter, so you get the initial priming first.
That's perfect. Other question, Abhishek Chakrabarti, former graduate at the Farber and now at Cleveland Clinic.
I have a question about
Go ahead.
The question was about the B7 family, general about how it's regulated. And so what caught my attention was you said that it appears as though the HHLA2 expression and the checkpoint inhibitors are segregating somehow in terms of the subset of so one is you look at this massive amount of cross are these receptors also expressed on the same genomic loci? Why could inhibition of one impact expression of the other? And then related to that is the concept of co-activation of both and therefore evolving.
Well, we found PD-L1 is strongly upregulated by interferons. And there's one of the things that happens is as the immune response starts, the T cells make interferon and that upregulates PD-L1. And so this multifunctional cytokine can also inhibit the immune response. And so I think that's the major pathway for upregulating PD-L1. But this pathway doesn't at all upregulate HHLA2. It's very different. We don't understand what does upregulate HHLA2, and that's really a very active research area because it's upregulated in vivo, but if you take the tumor out, it loses it pretty quickly. So there's really one of the critical questions now is what is it in the body that upregulates these immune evasion signals, which that signal isn't there in a tissue culture plate. It's not hypoxia. We've eliminated that, but really finding what it is really an open area now.
I mean, perfect, perfect. Any other question here? Dr. Tanir from MD Anderson, these are?
It's inspirational the two our couple have done here. So I have a son who's about 10 and really interested in immunology, following the footsteps. So my question is actually related to what Lisa asked earlier about the field. There are colleagues of yours who believe that if we just understood the intricacies and the workings of the immune system, we will probably won't need chemotherapy, won't need target therapies. Do you believe, I mean, we're at a point where once you really know all the molecules on the B cells and the B cells and NK cells, all those things, once we know the immune system, do you think we will be able to cure cancer, to get to the 50% or maybe 100% CRA cure for every patient with cancer, regardless of their tumor type or the molecular underpinnings of that cancer?
Great question. So, that's a politically correct answer because a lot of the oncologists are not immunotherapists, So do you really think at some point, and it's a bit of a philosophical question, in 5-10 years, we'll understand the immune system in a way that there will be way more options that are chemotherapy-free, targeted therapy-free? And I don't want to put you on the spot, but where do you think, if you have to predict, so we can have a 50 and 100% CRA?
I think that's definitely coming. I think we all realize now the door is open and that there are real possibilities. And I think this is attracting the best, most creative minds around, and tremendous energy in the pharmaceutical industry. And the tools that are being developed in terms of genomics and protein and drug constructs are just almost magical. That just this multiple of positive advances is really going to make cancer much more curable.
I think the other thing is that we're beginning to get an interdisciplinary understanding. The immune system isn't acting alone. For example, there's a lot of recent, very interesting work of neuroimmune interactions and how those interactions can regulate immune responses in different tissues. Many of our tissues are innervated, and there's crosstalk between the immune system and the nervous system. So I think when we think about where immuno-oncology is, it's really bridging two disciplines and bringing people together. And I think that achieving more of an integrated understanding, also bringing in our understanding of neuroimmunology as just one example, is going to help us understand the regulatory cues in different tissue microenvironments. And that's going to help us get to the next level.
Just a follow-up question on this. So obviously, so that's the goal. And I would say, what I'm hearing a little bit is, we do all these multi-omic studies. We can look now at almost any proteins, any genes, expressions, etc. But it sounds like what you two have done is really do deep science, right? Where you took a certain pathway, a certain family of molecules, you really drilled deep into the science that allows you to maybe develop these better, further, and get more insights. And how do we keep a balance, so to say, not from a funding perspective, so to say? So let's say, from the DoD perspective, how do we utilize the funding the best way to keep a balance of the deep sciences needed in drilling down in some of these more exciting pathways and these expression studies that we now deploy so widely in clinical trials?
I think that's a great question. And to me, I want the results from the clinical and the omics studies because it helps me understand what to focus on. I want to work on translationally actionable, molecules or pathways. Understanding the PD-1 pathway very deeply. I mean, it's really more like a network now because you have PD-1 interacting with PD-L1 and PD-L2. PD-L1 also interacts with CD80B7 and PD-L2 interacts with RGMB. So understanding this network deeply, how it affects various types of immune cells at homeostasis. This pathway has an important roles in regulating tolerance and resolution of inflammation. And if you think of a tumor as a wound that never heals, that we really, can learn a lot by understanding this pathway very deeply and what it interacts with.
Perfect. Perfect. We have a question from Dr. Voss from Sloan Kettering.
Yes. So I have a question. I think I'll just speak up. So my question is coming. We keep coming back to the thought of combinations. So the dynamics of the microenvironment that you keep pointing to, and I wonder how your deep understanding of this network might help to help us think through clinical trial designs. So what we have been starting to do in kidney cancer to speak to the heterogeneity of disease is to think about adaptive clinical trial designs. And there are some trials that have been conducted and presented in that space. But the approach has always been for adaptive designs to have sort of a black and white approach. You try something, and if it doesn't work, if there's progression, then we randomize and add in ipilimumab, or we stop the approach and so forth. And I wonder if we better understood the microenvironment the way you pointed out, could we adapt not so much to abandon a strategy, but just to tailor it or sort of hone in on a response? So do you see any way in the near future where we could have adaptive designs where we actually, through peripheral blood analyses or repeat biopsies, can see how the immune microenvironment is changing based on drug A that we gave, and then maybe give a second checkpoint inhibitor and so forth? And I wonder if there's any models that come to mind or any targets that come to mind that could be applied in kidney cancer.
So I think one of the technologies now is that one can use, for example, the T-cell receptor as a molecular barcode. So you can look in the blood over time and monitor changes, and then have the opportunity to look at the tumor and the lymph node at the same time to understand how things change, to understand what is changing when there is a response, what is changing when there's a lack of response, and studying that as well. So I think that that offers the same way that one can look at tumor evolution, one can look at immune evolution.
And that would be great. I mean, to look at, at some point. Any, questions coming here? That's great. Dr. Kutikov from Fox Chase locally.
Well, great to see everybody in Philly. Welcome. So I have a sort of off the wall, random comment, an idea that I think really hasn't penetrated oncology, but has really sort of dominated some of the work in the law enforcement community. I mean, the law enforcement folks have gone to, 23andMe and those giant databases of basically genetic sequencing that's been done in the community, and have solved some of these crimes that have happened 30, 40 years ago. And that's really sort of gotten a lot of traction in the popular press. Some of these trials that some of these crimes have been committed in the 70s have all of a sudden been solved by tapping into these databases that are just incredible. I mean, I think as an oncology community, it's kind of sitting in our lap here, and nobody's really tapped into it, unless I'm not aware of it, of some of these answers that may be lying right around the corner. These patients that have been responders to some of these immunotherapy agents that are, or even IL-2 responders, and it is very sort of within reach to tap into these databases and see how these, what are the genetic changes, what are the signatures that are very, sort of there's similarities between their relatives. For instance, the idea here is, even if you haven't donated to 23andMe, which I haven't, but apparently even if your third cousin has donated, basically the law enforcement community can track you down now. No, it is really incredible. I mean, there's just basically just the small percentage of folks have donated their genetic material to these databases. Basically, all of us can now be reached. So I think just a random idea to the community there. Maybe if somebody can access these databases to answer some of these questions, I think there may be an opportunity there.
Well, I'll take that because I have a lot of fun on ancestry.com with my DNA relatives.
That's great.
There's clearly a lot of privacy considerations there with the 23andMe or Ancestry, which would concern a lot of people. Nevertheless, I think we're coming towards where we're going to sequence every patient's tumor and be able to see what the mutations are in that tumor. And knowing those, an individual sequence is going to let us identify the best targeted kinase inhibitors, which will work on that patient. And it'll tell us what the mutations are, which would be good tumor antigens, which people could use in vaccines. The cost of doing a sequence is coming down drastically. The actual data in like 23 and Ancestry is actually it's a snippet of information. It'll tell you whether you're related, but it won't give you deep understanding. So actually those databases don't have the information necessary for patient choices.
Yeah, I'll push back a little bit. Apparently apparently the sequencing that the FBI has is it's just some SNPs, but the 23andMe is sort of the entire genome. Just putting it out there, maybe an opportunity for us as a community to look into this and partner with them. Because if FBI can have that data, I think we can have that data. But
I'm not sure about that.
I'll ask a real question here.
Yeah.
Why do you think there's a difference between the agents that block PD? The simple kind of surgeon question that always I've never heard the answer that was satisfying. But why do you think there's a difference between blocking PD-L1 versus PD-1 versus the ligand? Why do you think those agents differ in clinical practice? Because they do.
I think there's a combination of two reasons. One is that there are two PD-1 ligands, PD-L1 and PD-L2. And PD-L1 is the major one, but using a PD-L1 antibody doesn't block PD-L2, so it can still be active. So that probably contributes some. The other is that PD-L1 has a complicated set of interactions. It doesn't interact just with PD-1. It also interacts with the B7-1 or CD80 molecule. And that then interplays with the CD28 CTLA-4 pathway. If PD-L1 and B7-1 interact, you can still get a positive stimulatory CD28 signal, but you reduce the inhibitory CTLA-4 signal. So blocking the PD-L1 also will block those interactions. So they're not equivalent.
And we do have, trial that tried to combine so far without major success, PD-1 and PD-L1 inhibitor for that reason. Actually, we're gonna take one question, then a final, question, because we need to wrap it up. So one question that came online, since only we took, a few questions online, from Dr. Lee Ellis at Cedars-Sinai. He's asking your thoughts about combating cell plasticity, not much heterogeneity. So it's both tumor and tumor microenvironment, and this could be an escape and resistant mechanism. So cell plasticity. Anything to say about that from an immunological perspective?
So we know that environments change over time. We also know, for example, some of the work that we've done with Marcia Heiges's lab is that with obesity, that there's a competition between the tumor cell and the immune cell for nutrients. And that it's hard to believe, but what we learned is that the tumor cell is able to starve the immune cell in those microenvironments because it's able to have different kinds of transporters and being able to then take away the nutrients, and that one needs to think about ways to restore them. In the same way, one can think about a tumor microenvironment and how each of the elements is affecting the immune cells, either directly or indirectly. And what we need to understand is how that evolves over time. The tumor is growing, the nutrients are different. What is different during the primary tumor, during a metastasis? And it's really, I think, an active area, an important area to learn more about.
Perhaps, you know, to wrap it up, one of the things people ask me is, you're in 1 minute. There's a lot of folks here that are, you know, postdocs, junior attending, fellow starting. If you want to give them each in 1 minute an advice, and they're all hopefully gonna go and cure kidney cancer. No, we'll cure it before, but they're going into kidney cancer research. So starting, Arlene, an advice for the next generation?
So I think several things. The first is mentoring, that I was really blessed to have incredible mentors. And I think it's important to have mentors that you can work with. I think all junior faculty and postdocs should have mentoring committees that can be sounding boards and be helpful to you along the way. That you need to know what to do when things are working and when things aren't working. And don't be afraid to reach out to others. I think mentors are very important. And then ask important questions. I think the way we've been talking this morning, thinking about how you can work and do something unique, maybe at the interface of two fields, interdisciplinary science, to move forward in a new way.
Perfect. Gordon?
Well, I think one of the great strengths of the kidney cancer community is its interactions. Its interesting to immunologists was that IL-2 worked in kidney cancer. Not a lot of patients, but some. And some of the pioneers of that advance are really associated with this group. And this made the community really interested in immunology. And it has been very welcoming. It's welcomed basic immunologists into the kidney cancer community to collaborate and contribute our understandings of ideas. So I think the advice would be to reach out, collaborate, bring multiple lines of attack into your work.
What a wonderful finish. So Arlene and Gordon, thank you so much for coming. Thank you so much for sharing your time and your knowledge and wisdom with us. And we're looking forward to a wonderful rest of the meeting. Thank you so much.